let's talk about the ear and hearing. The ear is basically divided up into three sections. The external ear, which includes the pinna, also called the auricle, and we said auricular means ear-like, so this is that elastic cartilage area that actually captures sound waves through these little grooves and stuff and channels them down the next part of the external ear, the external acoustic canal. And of course this is lined by what we call ceruminous ducts, because this is where we produce this stuff called earwax. And then we have the boundary of the external ear, which is the tympanic membrane, which sometimes is also referred to as the eardrum. Then we have our middle ear, which on one side has the tympanic membrane, but it contains our ear auditory ossicles, which are our bones inside of our ear, which is the malleus, incus, and stapes, or altogether we call them the auditory ossicles. And it also contains our eustachian tube. Okay, that eustachian tube is necessary for us to be able to equalize air pressure between the throat area and the external portion of the ear. Then we have the inner ear area, and that is has a region or has an area called the bony labyrinth, which is a structure that has three major parts to it. And we also have one of our ear ossicles attached to that bony labyrinth. So that inner ear area is, of course, encompassed with bone. It's in that petrous portion of our temporal bone. Okay, now looking at these ear ossicles, notice that I have a bone that's actually attached to the back of the tympanum or tympanic membrane, the eardrum. It's called the malleus, and that is attached to another bone called the incus, and then that's finally attached to a third bone called the stapes. Now notice on this we actually see little ligaments and muscles that are attached to some of these ear bones, and what that basically becomes is an amplification system. So if we hear very low sounds, very low, quieter sounds, then these muscles loosen up so the bones can vibrate more. If we hear really high, loud sounds, then the muscles can tighten up and thus tighten up the bones, and so we don't shake the bones as violently when those air pressure waves hit our tympanic membrane, because when we vibrate the tympanic membrane, then it vibrates all three of those bones. Okay, next we have this area called the bony labyrinth, and notice it does have three regions to it. We see the snail-like region called the cochlea, and that actually contains our organ of hearing called the organ of Corti. We'll talk about that in a little bit of detail later. Notice that we also have, on the opposite end, we have areas called semicircular canals, and these semicircular canals, there are three of them because they're basically monitoring our three planes of space, because we live in a three-dimensional world. And then between the semicircular canals and the cochlea, we have an area called the vestibule, and it has some structures in it that are very important for balance. Okay, and in our sense of direction. Now, in looking at this, notice that we have a bony area, which is outside. Then we have a blue membranous area, which is inside. And between the outside bony area and the inside membranous area, we actually have a fluid called perilymph that exists just underneath that bony area, but outside of the blue area. And that's a very watery, aqueous fluid, very similar to cerebrospinal fluid. Now, in the membrane, in this blue membrane, we actually have a more viscous, thicker fluid called endolymph, and it's more similar to intracellular fluid, and it flows inside that membrane. So I've got a more watery fluid outside the membrane, but inside the bones. I've got a thicker, more viscous fluid inside the membrane, and these fluids interact with each other, and we'll see that in just a few minutes. Okay, looking at the cochlea first and trying to describe what's going on with how sound is created. In this picture, what we see is the movement of sound waves through the external acoustic canal. We see that it vibrates the tympanic membrane or eardrum back and forth. Now we have our malleus, incus, and stapes all attached to each other, with the malleus being attached to the tympanum, the incus being attached to the malleus, and the stapes being attached to the incus on one side, but being attached to the bony labyrinth on the other side. And it specifically is attached to a membrane called the oval window. Now, if we understand and we look at that And what we see on the right side of the picture is we actually see the cochlea that's been partially unwound. Now notice we have three regions within that cochlea that we call ducts. The top duct, or the first duct, that would be connected with the oval window is going to be called the vestibular duct. And again, we have that fluid called perilymph in there. Now the middle duct, which is the cochlear duct, and that's where our actual organ of hearing called the organ of chordae is found. It also has that membrane that surrounds the organ of chordae, and so it's going to have endolymph, that thicker fluid in it. And then finally, the last duct, which is called the tympanic duct, has more perilymph or watery fluid in it, and then it actually ends at an area called the round window. So when we vibrate, when the stapes is vibrated by the other bones and by the tympanum, then we vibrate the oval window. That sends a wave through that fluid, which will eventually cross over the middle duct and the middle membrane, causing it to vibrate up and down. And then the wave will eventually go into the round window, where it will push out the round window and resolve itself. Now, one thing, the lower the sound wave, the higher up on the cochlea the wave it'll actually go towards the apex or the top of the cochlea. The higher the frequency, then it will actually cross over sooner than it is in this picture. So if we had a lower frequency, it would cross over later than we see in the picture, and the highest frequencies would cross over at the apex or top of the cochlea. The lower frequencies would cross over closer to where the oval window would be, closer to the round window. So the highest frequencies we could perceive would cross over there. The lowest ones would cross over later on. All right, looking at that cochlea, notice again how we actually have our three chambers. We do a cross-section of the cochlea. If you look at either the lower left-hand side of the picture, or even see how the cochlea is arranged in the upper portion with the stapes on it. But nonetheless, what we want to notice is that we have three ducts with the upper and the lower duct having perilymph in it, the middle duct containing the organ of chordae, and also having the more viscous endolymph inside of it. Now, that organ of chordae in that middle duct, in that cochlear duct, is actually the organ responsible for hearing. So basically what happens is when we push that wave of fluid from the vestibular duct, the upper duct, across the cochlear duct into the lower duct, the tympanic duct, and what we do is we cause part of the organ of chordae to move, and the part that moves is actually called the basilar membrane. It can actually vibrate up and down. Now notice in the basilar membrane we got support cells and we have hair cells, both outer hair cells and inner hair cells. What happens is when the basilar membrane vibrates up and down, then the top of the hair cells, called stereocilia, actually makes contact with a rigid, stiffer upper membrane that can move some itself, called the tectorial membrane. If we make contact with the upper membrane from the hair cells, and what that does, that elicits our nerve impulse, which we were just now learning is called an action potential. Again, here we can see in this picture just the organ of chordae itself, which lies totally in that middle duct, the cochlear duct, and we see that again we have the tectorial membrane, the more rigid membrane. We have the basilar membrane, the bottom membrane is more flexible, that actually flexes up and down when the wave goes across it. We see the hair cells that have nerve endings attached to that, and we see the stereocilia on the end of the hair cells, which would, once the basilar membrane bounces up, they would make contact with the tectorial membrane, and when they make contact, they actually cause it to send a nerve impulse, an action potential. Okay, here we see three different examples of. We notice on the left-hand side we see the stapes, where our top duct is, our vestibular duct. Notice that in the top picture we have the different frequencies. Notice the higher frequency, 16,000 megahertz. Then later on we have 6,000, and later on we have 1,000, because when we send a super high frequency, we actually cause the basilar membrane closer to the stapes to actually vibrate up and down, and therefore the wave would cross over that middle blue area to the lower tympanic duct and bulge out the round window. Now in this particular example, going from A to B, notice that the basilar membrane is actually detecting sounds around 6,000 megahertz, and notice that from B to C. Notice how that membrane bounces up and down, and that our round window actually gets pushed out and then back in. It's kind of like a recoil method, but that's going to detect sound within that 6,000 megahertz frequency. Okay, what happens to that sound? Again, our low frequencies go further up our apex, high frequencies are found further down, and we have nerves that are detecting each of those. And notice they're going to go back into the brainstem on cranial nerve number 8, and then from the brainstem, we're going to actually synapse onto the inferior colliculus, part of the midbrain or mesencephalon, and we're also going to go ahead and synapse up to the thalamus, and of course from the thalamus we send a synapse to the temporal lobe, where we actually interpret the different frequencies of sound and put all that together to interpret what we call sound. Notice again the high frequency are interpreted further in on the temporal lobe, the lower frequencies are actually interpreted further out on the temporal lobe. Okay, now next we have actually our vestibule, or the spaces between the semicircular canals and the cochlea. And notice that we have a big membrane area called the utricle, and then a membrane called the saccule. And within the utricle and saccule, we actually have structures called maculae. So those maculae are actually detecting different types of movement. The maculae is sensitive to up and down movements and to linear and straightforward changes in speed, and that's actually called static equilibrium. So if you've ever been in an elevator and felt that sensation of going up or down, or even been in a car and felt the sensation, even if you had your eyes closed, of going forward or backwards, then these were being activated. Notice that the maculae itself actually has hair cells, similar to what we saw in the organ of Corti. They actually have stereocilia that's actually embedded in a gelatinous material. There are support cells around it, and then there are nerve fibers on the hair cells. Now, we have some calcium carbonate granules called otoliths that actually are on the, and together they're called the statoconia. And notice that this otolithic area is on top of the gelatinous material. So gravity causes the calcium carbonate granules to shift, and when that shifts, it actually shifts the gelatinous material, which then bends the stereocilia, causing an action potential to be sent through the vestibulocochlear nerve, or cranial nerve number 8. Okay, again, you can see the stereocilia coming off the hair cell. You can see the nerve endings that surround that. So if we move those stereocilia displacement, then it actually causes the sensory nerve to send the message. Okay, notice in the picture, when his head's in one position, the gravity is causing the statoconia to stay in one place, but when he moves his head where the gravity causes the otoliths to actually move the gelatinous material, then the hair cells actually have their stereocilia bent, and therefore it fires off action potentials, kind of giving us the sensation of movement. Again, we can see these two factors, again, controlled by gravity, tilt of the head, or movement, which creates the gravity. Okay, next we have our semicircular canals, which are 3 fluid-filled canals, and they're oriented in the different planes, so we know we live in a 3-dimensional world. So we have these areas called ampulla, these large bases of the semicircular canals, and we see that we have our membranes in there with our indolent fluid, and then we have some structures that called crista ampullaris, which are the structures that are within each of those enlarged bases called the ampulla. If we look at the crista ampullaris and look in the ampulla, we can see that the crista ampullaris has, basically, it also has hair cells with the stereocilia protruding into a gelatinous-like material called a cupula. We have the support cells around it, and again we see our sensory nerves that are attaching to the hair cells. Now, whenever we move in one of those directional planes, then the fluid is kind of like if you've ever used a level. And if you use the level, you've got some fluid and a bubble in the middle of it. You try to line the bubble up between 2 points with the way you move your level. So we have a very similar thing going on